Okay, good morning. Thank you all for joining us. We are going to jump right in, introductions in sort of a rolling fashion as we get the conversation going. I've learned in past years that the allotted time for this session or any of these sessions is always shorter than we might like. I'm Keith Flaherty, I'm the moderator. I'm a medical oncologist at Mass General and focused on therapeutic development throughout my career. I thought for this discussion today, I thought it might be timely to talk about sort of two sort of paradigm shifts in oncology, at least in academic medical center oncology, where we are wanting to shift our sort of treatment or point our treatment armamentarium at earlier and earlier disease states. Not a brand new concept, but like leveraging emerging technology essentially to do that. And at the same time, we are constantly trying to expand our therapeutic armamentarium, so discover new drugs. But what I thought would be maybe useful for today's discussion is to really collide those two concepts, because we're attempting to do that, certainly at Mass General Brigham. Other major academic centers are as well. And so I assembled a group of friends who I know can handle this topic. Basically, you know, two of the panel, leaving myself aside, are card-carrying academics presently, which is to say reside in the same tall ivory tower that I do, where we'll talk about this sort of collision of science and medicine and kind of what might be possible as a way of trying to ultimately improve outcomes, but discover new therapies along the way. Brought along two friends who, of course, both have academic backgrounds. Who doesn't? But have a foot in something more like the real world, as in the private sector, and have advanced diagnostics and therapeutics in the real world, and ultimately that's going to be quite relevant to what we're going to talk about today. Like, what is the evidence we need to generate to actually create a paradigm shift in terms of intercepting disease earlier, applying new therapies, even investigational therapies, in earlier and earlier disease states. So that's kind of a bit of the framing of what we're hoping to accomplish today. So I'm going to have each of our panelists who are listed here by title sort of self-introduce as we kind of get rolling into the conversation. So I thought maybe Aparna, I would sort of have you start us off again from the tall ivory tower. What I've framed so far is, in some senses, not totally new. The idea of like introducing therapy earlier and earlier in the cancer disease course, like sort of going from patients with metastatic disease to deploying therapies in the adjuvant setting and witnessing, cures with the same therapies that can only extend survival or palliate patients in the more advanced setting. That's an old concept. Detecting disease with molecular tools is actually not a very new concept in hematologic cancer. It's just a much more new concept in so-called solid tumor oncology, which is 80% of cancer. So maybe if you could just maybe take a run at sort of, what's changing, what gives us reason for optimism that we actually could begin to extend, what's been done to a reasonable degree in hematologic cancers, but now in solid tumors based on sort of technology advances.
Great, thanks so much, Keith, for having me. So I'm Aparna Parikh. I'm one of the GI medical oncologists at Mass General, and I run our colorectal cancer program, but have also been really involved over the last decade in this concept of molecular residual disease or minimal residual disease. So this idea that you can detect various blood-based analytes to detect microscopic levels of tumor before you actually see something on a CAT scan. Our group has been very involved with some of the early clinical data generation in this space, But to take a step back, I think, as Keith is alluding to, the problem with even curative intent solid tumors is that around 30% of patients, even if you look at the top 3 malignancies, colorectal cancer, breast, and lung cancer, will still have their cancer come back. And historically, when we have done sort of adjuvant trials, again, therapies after curative intent treatment, it takes a really long time to actually provide the evidence that a therapy is working. But with these new technologies, and again, I'm talking primarily today around what's called circulating tumor DNA, again, the ability to detect a small amount of tumor in the blood, but there are many other newer assays that are also being developed. But this idea is that if you can detect now, even as low as 1 part per million of circulating tumor DNA in the blood, you can pretty much have confidence with nearly 100% positive predictive value, not quite 100%, that your cancer is going to come back. And the time from detection to the radiographic or clinical recurrence is give or take a year, some a little bit less, some a little bit more. So that presents a unique opportunity to shift the paradigm from testing drugs in the sickest patients, the therapies where therapies are least likely to work. Patients by this point have been exposed to a lot of therapy, have many alterations that may drive their therapeutic resistance. And the opportunity certainly to cure is no longer there. And what we see in current drug development is median survivals of just a few months, and those are unfortunately our game-changing wins. And so can we take that traditional approach and use these novel biomarkers to intercept early with the goal of curing more patients?
Okay, so Vince, I'll maybe turn to you. There was a time that Vince actually did work for Foundation Medicine, as this sign says, but he'll update us. I think maybe the organizers were thinking they would channel your years ago leadership role in Foundation Medicine, which was one of our real pillar companies advancing particularly next generation sequencing broadly, really nationwide, at a time that we academic centers thought this was ready for prime time in terms of clinical practice and to drive research as well. But there were real headwinds, and Foundation Medicine, more than any other company, really pushed significantly through those headwinds. But you have a variety of career experiences beyond that role at Foundation. I guess my question to you is, what are the not what the problem is with what Aparna just said, but what are our challenges in terms of trying to establish evidence, that finding disease, residual disease, and treating it actually results in a bona fide clinical benefit. And when I use that term, ultimately mean that from the perspective of actually ultimately convincing our colleagues at FDA, for example, that we could demonstrate evidence of clinical benefit in a setting like Aparna is describing, and actually start even potentially getting drugs approved or regimens approved in that setting. So maybe help us think through the how do we actually extend this paradigm in a way through investigation to generate evidence that actually could shift the field.
Thanks, Keith, and thanks for inviting me. I wish it were 14 years ago in some ways, but to that end, I'm unaffiliated now, and I have a feeling, as I look at sort of my worldview as someone who worked in lung cancer for many, many years, that we've had these eras of therapeutic plateau. We sort of had that in the checkpoint inhibitor space now in therapies. I feel as if some of the tools that we'll discuss today are enabling us to anticipate the next step forward, and particularly some of the technologies that I see in the diagnostic space that really allow an integration of spatial, functional, cell type, along with quantitation of not only the tumor in toto, meaning both the tumor microenvironment, right, which can be 50, 60, 70% of a tumor when you look at a biopsy, and the remainder of the actual tumor cells themselves. So some really powerful technologies coming both on the DNA, RNA side, and some of them can be also enabled to be MRD not only detectors, but not only let you know that you are dealing with a cancer, but to simultaneously, in many cases, characterize that cancer, which is, you know, detection is necessary, but it's not sufficient in some ways. And so that's pretty exciting. I love the idea of agnostic platforms. That was one of the attractants of going to FMI. Let the best drugs win, the best companies win. It was really, let's tell patients and doctors and researchers and biopharma with what they're dealing with, let them figure out how best to move forward. And so to Keith's question, you know, one of the early lessons there was you do need some biopharma champions that, and that can be a little hard in the MRD space, right? Because it takes so long, perhaps, to do trials. There have been some interesting studies in hematologic malignancies lately, which, you know, which have potentially shown the ability to, that MRD detection treatment matters. It's not simply a case of lead time bias, which will always be the, you know, the first pushback from any reviewer, et cetera. So those are just some high-level thoughts. I think MRD is a little bit more challenging than, say, doing things in advanced disease from the standpoint of getting the right trials initiated and robustly supported. I think it might be a bit more on the liquid side in some ways, but I suspect, you know, Aparna and others may have more creative thoughts than that as to how to do it in the more common solid tumors.
Great. So, Mike, we're ultimately talking about developing therapies or deploying therapies, but also developing therapies. So, I think what Aparna and Vince have both framed is the idea that we potentially are on the verge of being able to tell a patient who's been diagnosed with what we think might be localized disease, offer them definitive therapy, surgery, plus minus radiation, and let's presume in a cancer context where there is some kind of systemic adjuvant therapy to offer. And we offer all of that, and we give all of that, and then after 2 months of that treatment, the patient is still MRD positive. So we've given them the conventional adjuvant therapy, and we're telling them you still have a problem. We can't see it on scans, but as Aparna said, like more and more evidence suggests like this cancer is going to recur. So you know, what's the tipping point to actually thinking about deploying emerging therapies? Take any of the assets under your watch or in your pipeline, and basically building, you know, an early evidence base that you could clear a minimal residual disease, and take that at least internally as confidence boosting that your therapy might actually be moving the needle, and then kind of maybe also help us think through, like from a regulatory perspective, like, you know, have you actually accomplished anything, you know, with that approach? And all of this, by the way, recognizing that our detection abilities, as Aparna touched on, have gotten better and better and better, like rather rapidly in recent years, and we all think they're going to keep getting better. So we're probably not done yet. This is a like shifting landscape of like how, like the threshold level at which we can detect minimal residual disease, and then this clearance question is kind of a related one. But how, as a therapeutic developer, you know, kind of how do you think about the squishiness or solidness of this, you know, as a foundation for developing therapies?
Thanks, Keith, and thank you for having me on the panel. My name is Mike Mim. I'm a pharmacologist at Astellas Pharma, and I'm currently the asset lead for PADCEV, which is an antibody drug conjugate that is indicated for the treatment of bladder cancer. And everything that you're talking about, Aparna, is very relevant for our clinical development program for PADCEV right now. So PADCEV is indicated in combination with Keytruda for newly diagnosed metastatic bladder cancer. But we've also been running trials and actually just read out a trial, Keynote 905, which is looking at perioperative treatment with PADCEV plus Keytruda in patients with muscle-invasive bladder cancer, so it's pre-metastatic. And we have just recently read out results, and what we see is that this combination, if given neoadjuvantly and then adjuvantly, actually improves event-free survival and overall survival compared to just surgery alone. It's well established, I think, now that ctDNA is a powerful prognostic tool in understanding how patients will do, how to utilize it to dictate therapy and what therapy to utilize in the adjuvant space and when to do it, I think is still an open question. It's a challenge. I think, to your point, Aparna, one of the key challenges is just how long those studies take, how large they have to be. But we're certainly investing and measuring in large global trials in this setting. Yeah. I think the challenge is, when do you kind of transition those from exploratory endpoints to your primary endpoints where you're really demonstrating and you can present that evidence to FDA. I think that work is probably in front of us.
Yeah. Aparna.
Yeah, and I mean, there is, precedent in the myeloma world. So, last year, I think through pooled analysis of over like 20 years of data, the FDA did acknowledge MRD as a surrogate endpoint. And, what we have tried to do is, as you mentioned, these trials are really, really hard to do, and kind of working in silos is just no longer going to be sufficient to try to create the evidence. So we have kind of put together, in the lens of a sort of nonprofit, a consortium that is really trying to bolster the clinical utility data generation. And we have, some folks from the FDA as well as biopharma and diagnostic companies all a part of this. But this idea is that I think the FDA is seeing that at least clearance can be used as a biomarker, not yet a surrogate endpoint, but that is, I think, ultimately the goal. And what is the data that we need to generate now that will allow that to ultimately be a surrogate endpoint? And what we're seeing for many of these solid tumor clinical trials is, DFS is still your primary endpoint, but you're using ctDNA clearance as either dual primary or a secondary endpoint. And in terms of the timing question, I think when your lead time to recurrence to your event is fast, you no longer need to wait 5 years for your recurrence event, because if you're positive, can you power your statistical analysis around a shorter time to recurrence? And you'll know quickly is if you cleared ctDNA, is that clearance real? Because you'll have your readout for those for the positive patients fast. And then if you can, push out the negative patients from recurring, I think that is giving you a signal in the right direction that you are actually not just kicking the, can down the road a little bit, but actually, hopefully, truly improving DFS.
Yeah, I think the point you're both making is that what we're talking about really is, I think, like sort of two shifts in paradigm. One is establishing a new cancer stage. So we used to just stage based on pathologic assessment and scans. But now, like taking blood-based detection, we're talking about sort of how do we qualify a new stage, sort of sub-stage or intermediate stage, really. And that's not something that a given drug developer or sponsor can do in the eyes of FDA or the field, right? It is a field-wide challenge. But generating the therapeutic benefit evidence is a new surrogate endpoint, as Aparna was and Mike both, I think, were just saying. So one paradigm shift, new cancer stage. The other is, can we actually create an intermediate endpoint that gives us much earlier, more confident surrogate information regarding true clinical benefit, sort of using FDA language here. Of course, we've used, in the metastatic setting, we've used response rate for our own purposes as academic investigators and sponsors as well, for kind of gaining early confidence. And there are even instances where the FDA has considered that to be an adequate surrogate of clinical benefit, at least for accelerated approval purposes. This is analogous to that. I would argue there's reason to believe this actually could be far better than our somewhat crude methods of measuring objective response with imaging. That blood-based detection gives us a little bit more of a something like more of a linear kind of response. Mike?
Just to add one quick thing, totally agree with everything you said, is as we're moving these combination treatments earlier and earlier in the disease state, the concern around overtreatment and toxicity is so
Yes,
important.
yes.
And so I think these are the types of tools, they're powerfully prognostic, but how we're going to utilize them to dictate what therapy and when for which patient, I think is going to be critical. Yeah.
So let me just pause to thank a couple of the folks here for posing questions through the app. I mean, I think if you've been in other sessions, you know that's kind of our preferred method. And I tried to grab the first of those questions already and weave it into one of my earlier questions to our panelists. So keep them coming. Got an excellent second one, which I'm going to come back to in a moment. But I want to maybe build from what you're saying, Mike, to sort of expand the aperture a bit and ask Liron to kind of help us think through this whole sort of, paradigm shift, earlier disease state, not just for deploying therapies of well-established benefit or somewhat established benefit, let's say in the metastatic setting, and now we start moving them further, but actually like really shifting our investigation of new therapies altogether in earlier disease context. I'll maybe make this as a foundational comment since we don't have a functional diagnostics expert on the panel with us, but I work closely with colleagues who are in that space. There's been a bit of a slowly building movement of sorts to think about taking material that comes from definitive surgical resection and screening drugs in that material, so-called functional diagnostics, entirely analogous to how it is that we work up antibiotic sensitivity once we get diseased tissue in the laboratory. And so actually developing methods like that to deploy standard of care therapies, but like be able to understand based on like ex vivo response to drugs, what is a patient potentially sensitive to. That's its own area in terms of establishing the evidence base and ultimately using it to shift care. But skipping over that for a moment and recognizing that what we're talking about is in the solid tumor oncology space is around definitive surgery. So whether we give pre-surgical therapy or neoadjuvant therapy or post-surgical therapy, adjuvant therapy, in any case, if surgical resection is going to be done, I would argue we currently underutilize that tissue in terms of trying to understand therapeutic vulnerability to establish therapies and then particularly for investigational therapy. So let me put it this way. In a context like breast, colorectal, or lung cancer, a patient gets pre-surgical therapy, neoadjuvant therapy, and at the time of surgical resection, there's still viable tumor left. That's a problem, right? That patient has effectively treatment refractory disease. We can doubly prove that by attempting to give them post-surgical therapy and finding that they're still MRD positive. But can we use that tissue as a really powerful way of focusing in on unmet need? I mean, patients whose tumors are refractory to our best current systemic therapies, and use that as a starting point for actually trying to discover new drugs. And this is something I know that is one area that your work, Liron, has been pushing towards. So maybe kind of help us understand, like, well, what are the insights that you can gain, from that kind of focus?
Yeah, well, Keith, thank you for having me on the panel. My name is Liron Bar-Peled. I'm an associate professor at the MGB Cancer Institute. And for the last decade or so, we've been really at the frontier of early drug discovery technologies, which are very gratifying when they make it into the clinic and when you can see a benefit, especially as a more fundamental biologist. Maybe to take a step back and just kind of like level set with regards to like where we are in the field in early drug discovery today. And I think, we've become, again, as a field, exceptionally good at making drugs for things that we know how to drug, like GPCRs or kinases. We are exceptional at that. And I don't want to say that we're almost at the plateau stage, but it's becoming exceptionally hard to make better and better drugs. And at the same time, there's quite an opportunity because there are these additional cancer drivers which we simply don't know how to interrogate using traditional approaches. And I think this is where more technology, and actually not only newer technology, but where we start our investigational studies is exceptionally important. You know, again, just being tutored or mentored by Keith, models are models, right? They're never going to be the definitive answer for what is cancer. And, you know, is there a way that we can bypass, you know, your cell line models, your animal models, and just start drug discovery in patient samples? And I think the answer is yes, and it's exceptionally exciting to be at that stage, because what are we missing when we're not doing that? You know, for us, we think about the ways proteins look, the unique conformations that they take, and the partners or their friends, which we need to be able to target as well. And all of this is lost. All of this is lost when you start using the traditional approaches. And so here today, now in 2025, when we pair with clinical colleagues, we're enabled to be able to start these drug discovery campaigns in patient samples, and not only test existing drugs, but then actually recover new targets that we would just never be able to see before.
Yeah, and just maybe a quick add-on point there that, you know, for decades now, I've been making the point that we are ultimately trying to wrestle down a very complex disease that's going to require sort of, you know, multi-drug regimens that, you know, follow the precedent of HIV, but like massively more complex disease that, you know, comes from our own cells that have 20,000 protein encoding genes, and so the permutations and the complexity are enormous. But we are way behind schedule in terms of developing rational combinations. You know, part of my argument for the necessity of kind of moving to the paradigm that we're talking about and interrogating, you know, the real most problematic cancer tissue sort of systematically for drug discovery and development is to be able to really accelerate our opportunities for developing combination therapies, because ex vivo, you can screen a tumor sample with combinations of whatever sort, and then of course narrow down then the funnel of what actually might be worth pursuing in clinical investigation. There's an excellent question that came from the audience regarding pathologic CR. So not to like skip over that you know, endpoint, which is how the solid tumor neoadjuvant subsector got going in the first place in breast cancer, right? By developing the evidence base that pathologic CR actually was a surrogate for overall survival improvement. That was a very long campaign, but followed some of the similar themes of what we're talking about in terms of the field, like sort of organizing itself and therapeutic developers as well, towards generating that evidence. And what's, you know, the question really was kind of what are the lessons learned from Path CR that, you know, might help inform, you know, how we think about, you know, the concept we're talking about today. To really maybe throw that to anybody who's, you know, has like watched that development. Maybe I'll just make the editorial comment that I'm a career-long melanoma investigator, just as the like kind of little subsector of cancer type that I focused on, but my drug development interests are, you know, completely broad. But I will recall conversations that a group of us melanoma investigators had with FDA when we were beginning to investigate drugs neoadjuvantly in melanoma, and basically we said, okay, well, we're starting to see this bifurcation of outcomes with pathologic CR, you know, yes or no. You know, to what degree do we have to generate our own evidence, FDA, to be able to establish Path CR as an endpoint, surrogate endpoint in melanoma, or can we borrow from breast cancer and then, you know, rectal cancer and other tumor types where neoadjuvant therapy is becoming a paradigm and where Path CR rates are meaningful? And this is a public open forum conversation that we were having, so I'll just directly quote. They said, oh no, you're on your own. You've got to generate your own evidence that this is a useful endpoint in melanoma. And so maybe I'll, you know, kind of throw this at others in the panel to opine on. You know, God forbid we have to do this. You know, what we're describing in terms of, you know, shifting and adopting use of these sort of earlier or minimal disease, you know, detection platforms, and have to demonstrate across every cancer type by site of origin. That'd be a very long ground war.
Yeah, I'll make a couple of comments. One is, I think one of the things that's enabling some of this work is the amount of information one can learn from a patient right? Going very, very deep. This is not CBio portal. This is the next dimension of that, going really, really deep on each of those patients and enabling some of what I think you're talking about, right? So as far as PathCR, I'm of an era where they used to show me, we'd see x-rays from patients treated with neoadjuvant chemotherapy and have a great response, and then you knew that patients with a PathCR resection, this is work Niall Martini did 50 years ago, that was a really good thing. So what were we doing? We were declaring the cancer status by the outer limits of our technology, right? That was the absolute best we could do. But now we have all these tools, and to your point, they're a moving scale. Will be whatever, a log order better, and that's where the debate in a lot of these companies is, and so forth. And so what we don't want to do, of course, is have to revalidate that at every cut, right? Oh, we're at 10 to the minus, 10 to the minus 6, and we're at 10 to the minus 8th, and so forth. So I think this is the type thing. I don't know if our friends at Friends of Cancer Research have taken this on before, but it might be a project for them, especially since they're not here. So that's a really good not-for-profit, right, that does get industry, FDA, academics around the table, and has really been able to build some consensus. It still can go out the window when you go to meet with the regulators, but over time it does bring in some gradual traction and some common points and some references that you can go back to when you go in with your dossier or whatever of how you're going to approach a certain disease state. So those are just some thoughts, not a singular solution, but it did occur to me this is something that might be well served for that type of forum, because what we try to do in that venue is come up with things that we think are ready for prime time, but the regulators, the agency hasn't really taken on yet.
Yeah, well, I'm being somewhat tactical and kind of composing a panel like this. I don't think we're quite ready for FDA yet. So this is like I think we'll get there soon, but I think we have to get ourselves organized first between academia and private sector, diagnostic and therapeutic developers alike. And then start posing questions about exactly as you're saying, kind of, well, how do we reduce this to practice in a way that will ultimately move the needle with FDA? But Aparna, let me sneak in a question as someone who investigates novel therapies in patients. So, imagine a scenario where, as Liron is suggesting, we can use patient samples to understand potential therapeutic vulnerability. I'd say potential meaning like it's ex vivo. So like in vivo, of course, tumor microenvironments are a bit more complex in the intact tumor in a patient. But where would you think about drawing the line if you were to give a patient, conventional adjuvant therapy, let's say? For how long would you go, based on current evidence, if they're persistently MRD positive, right? Before you would then say, okay, we're declaring that a failure, because normally you might give that adjuvant therapy for a year's time, just based on the kind of, in the era in which I used to tell patients we're not smart enough to know whether you've got disease or not, so we're just going to give you this, therapy because you're amongst a group of people who have some known risk. But now, we're talking about personalizing, adjuvant therapy and terminating it and potentially saying, okay, you've actually exhausted our available therapies and willing to consider giving you an investigational treatment now.
Yeah, I mean, really, really important question. I don't think we entirely have the answer yet in terms of how long, which therapies, but I think what is really reassuring that we are getting feedback on even from the regulators is that, because of the specificity of this endpoint, that I think this idea that you can actually now test some of these novel therapies in this setting is evolving. I think there is, actually one example right now, the solid tumor trial using a cellular therapy in the MRD space. And if you would have asked me even a year ago, do we feel comfortable about giving a highly toxic cellular therapy into that setting, I would have said no. But I think people are seeing that, to Keith's staging point, I think this is metastatic disease. These are patients where the cancer is going to come back. And so I think the ability now to trust novel drugs is shifting, and not just, once you have a kind of phase 2 dose, we're even being able to see some of these therapies being used in sort of early signal seeking and even, dose escalation in some patients too. So I think that's exciting, and the timing, I think, is still to be determined, and, we give a lot of therapy 6 months, a year, we just don't know, but with these biomarkers, when you can actually follow levels, it's not just yes or no binary, and with these newer assays, you can actually get quantification. And so we're trying to be very systematic within this consortium of how you're following patients. I think to your point around if you used a first-generation test for one of these trials, like how do you make sense of a more sensitive assay? And so really being thoughtful around doing the biobanking at the same time points where some of these tests are being used. So I think to be determined a little bit, but I think an opportunity now to even take earlier drugs rather than waiting for kind of full approval and then testing it in this setting.
Yeah, thank you. And Mike, a question came about sort of circulating tumor DNA like decreases or even clearance, with just let's kind of most commonly used platforms currently, recognizing that more sensitive methods are coming. As a drug developer, how do you think about this issue of sort of sensitivity of impact on circulating tumor DNA? Considering that we have pretty large amounts of evidence emerging across therapeutic modalities, that if you look at tumor regression data, so-called waterfall plots, but using tumor regression and conventional imaging, and then in the same cohort of patients look at changes in circulating tumor DNA, time and time again what we see is sort of deeper impact on circulating tumor DNA than we see radiographically. Like, so patients who have stable disease, as was posed in the question, some of whom even have clearance of circulating tumor DNA. Like, should we be getting excited about that when we know that the vast majority of patients who have only stable disease in the fully metastatic, radiographically evident metastatic setting generally don't derive very long-term benefit from most of our therapies? Like, sort of, this has become a part of early clinical development, almost universally, to measure circulating tumor DNA, and like, to what degree does it actually really, inform us in terms of increasing, decreasing confidence or probability of success?
Yeah, I think it's all about confidence right now, and it's because, and I think it's very similar to PathCR, where it's one thing to have this evidence, it's another thing to have enough evidence to convince a regulator that you can make treatment decisions on it. And so I think that's where the field is. That's at least how my team perceives it. It is, we want to measure it. We believe that it can help. It's just, you have to get to that level of investment, I think, where you're really proving, for, in a randomized study, that it's changing the meaningful endpoint outcomes, and that's usually event-free survival, disease-free survival, or overall survival. Yep. So I think that's where it is in terms of making decisions on trials. We're certainly measuring it, yep. But being able to utilize it and then make treatment decisions with it, I think we're still short of that.
Yeah, I'm with you. But I think what you're also saying is we're gathering the data.
Of course.
And broadly across drug developers. By that being such a sort of common practice, one would expect that we'll be able to sort of pull that data and understand sort of where we are, at least based on sort of current technology, and then, like, again, technology will continue improving. But I also, so let's say that the sort of the data gaps there are going to be filled. Liron, you alluded earlier to kind of the misrepresentation of the model systems that we've always relied on. Immortalized cancer cell lines, notably, which, as a semi-quantitative estimate, I always say on a good day, they represent maybe 20% of cancer biology, broadly speaking. So I guess I want to task you with kind of maybe weaving together two intersecting areas. One is that problem, and the degree to which you're actually able to, like, sort of, screen therapies in various cell states. Because when we talk about circulating tumor DNA assessment, I think just about everybody, like, first blush, what they think we're talking about is measuring mutant alleles, ideally sort of truncal drivers that are, like, present in every single cancer cell in a patient, so not subclonal phenomena. So we're measuring disease at that level, and we're just watching sort of rising and falling. That's proven to be somewhat useful. But there increasingly are methods, and I would say evidence even, that you can infer from circulating tumor DNA more than that. So you can actually get almost like a inverse transcriptome type of insight that would tell you something about cell state, which increasingly we have learned under selective pressure of therapy, cancer cells will shift their state, or at least be the problematic states will be enriched. So thinking ahead to deployment of those technologies, where we can actually in blood even can start to say that a lung cancer patient with an EGFR mutation who's been treated with an EGFR inhibitor has epithelial mesenchymal transition type biology, and we need to direct therapies, screen therapies. Again, just kind of expand a little bit our horizon for how we think about this idea of like intercepting cell state.
Yeah, so I mean, that's a really fantastic question, and one that's been very close to my academic heart for a while. I think context matters, right? And as much as I bemoan cell lines, we use them quite a lot because you can use them, you can do experiments. Increasingly this can be done more in patient samples. But you're right, in addition to just single points of measurement with ctDNA, you're able to see a much larger swath of things. And the question is, can you better understand the activity or the readout of cell states, which we now increasingly can understand by counting different transcripts that we can readily detect in blood or even in tumors. And then the question is, can we now marry that, particular hypoxic cell state or a tumor that's undergone EMT to an appropriate level of drug? Today, no, but looking toward where the puck is moving, I think, yes, this is something that we're absolutely going to have to do. But it's just simply not going to be done the way that we're doing it today, right? Like where you have, I realize this is a more broad audience, but just to be incredibly granular, you take your favorite, oncogenic target, and then you study in it, just completely in the absence, or you try to develop a drug completely in the absence of the cell state. I don't know how that's going to translate into more of, a new molecule against a resistant state, right? Because resistance is quite complicated. And, the question is there a clever way that we can just bypass having to be able to model it? And I think, yes, to kind of bring it back, starting with patients and starting with patient specimens is the way I would go about it.
Yeah.
It's just, I think I made this point earlier, but it does beg the question. I think this is the quantitation is not sufficient, right? We need to be able to learn from what we find in the blood, the tumor state. So some of the tools I've seen, right, that characterize the tumor microenvironment, drug agnostic, tumor type agnostic, what's the ratio of the TME versus the tumor, and those sorts of things. That's waiting to be explored, characterized, and integrated into our interpretation and learnings. And in turn, there'll be some hits from smart folks who put it all together. But I think that's part of the next step forward.
Yeah, I think maybe what you both are touching on is that we've had a way of kind of procuring tumor specimens on the path towards trying to do, offer benefit to patients by doing definitive surgeries, and then processing those specimens where we kill the cells, basically, or kill the tissue, because that was sufficient to be able for us to be able to then do H&E sections, immunohistochemical analysis, and tell a patient you have non-small cell lung cancer. And I guess maybe I would wind back the clock to say, there was a time in the evolution of lymphoma diagnosis and therapy where if you undersampled the tumor and did a so-called fine needle aspirate, pathologists wouldn't be able to classify the disease. And so then the paradigm shifted, and where basically if you suspected the possibility of lymphoma, that meant you had to do a core needle biopsy, a bigger gauge needle, to get more material, which does potentially pose a little more risk in doing such a biopsy. But if you can't diagnose the cancer, then you're kind of you're dead in the water in terms of being able to then assign treatment to that for that diagnosis. So what we're talking about here is another change in behavior, to be clear, even at major academic centers. So to have a surgical sample be treated as living tissue and not allowed to just kind of hang out on the pathology bench for a bit of time, because ultimately it's just the cells are going to be killed and be analyzed, and DNA can be isolated from fixed or dead tissue in that sense. Live cell analysis is something that is quite a recent innovation, even in academic laboratories. For this to ultimately become part of clinical practice, I mean, like where you're actually making treatment decisions based on living tissue, that's another area where we have to generate, frankly, the evidence to support that this is actually important or worthwhile. I think we are at time, if I'm understanding our session schedule, but I knew that we'd be able to keep going way beyond. But thank you for the excellent questions that I hope we were able to kind of incorporate in our discussion. Thank the panelists for thinking on your feet, as I knew you would, and joining us today. And thank you all for being here and for your engagement in this session.